Hey guys, this is Audrey. This is my first time actually recording a narrated lecture, so we will see how it goes. Hopefully you guys can hear me okay. This is going to be lecture 8. We're going to be going over vitreous and lens. I put this picture on here because this is actually the eye that I dissected when I took the ocular anatomy lab course. So if you look, I wonder if my mouse works here. If you look here, you can see kind of the vitreous gel. You can see an imprint from the ciliary body and then the zonules that were actually holding on to the lens. And the lens was really cool. It was really hard. We kind of popped it off just like a Skittle, kind of skidded it along some print, and you could see that it actually magnified it. So it was actually really cool. Here's the sclera over here, and we'll go ahead and get started. Okay, so some facts for you guys. The crystalline lens. Okay, so the crystalline lens is about a third of the eye's refracting power. So if we say that the eye is approximately 60 diopters total, a third of that would be 20, so 20 of the 60 diopters is held within the lens. The lens is a biconvex structure. It's posterior to the iris and the pupil. You can see that here in this photo. It's also anterior to the vitreous, so vitreous is all back here. We say that the lens is 10 millimeters in diameter, and it's about 4 millimeters in thickness. Your lens actually grows throughout your life at a rate of about, let's see here, 0.23 millimeters per year. So let me shout out those numbers for you again, guys. The diameter of the lens is going to be 10 millimeters. That's actually from top to bottom. The thickness we talk about is actually kind of the width of it. That's going to be 4 millimeters. And then the growth rate is 0.23 millimeters per year. Okay. So we know that the lens is connected to the ciliary body via the zonular fibers. There's lots of different names. The suspensory ligaments, the zonules, all of that is synonymous. It means the same thing. Okay. So some more dimensions for you guys. We always talk about the radius of curvature for curved refracting surfaces in the eye. So for the front surface, the anterior radius of curvature is going to be 10 millimeters. Okay, and then the posterior surface is going to be 6 millimeters. So 10 anterior, 6 posterior. So the posterior is actually steeper than the anterior. Okay, so moving along, we talk about what the lens is composed of. It's actually only 40% water and 60% protein. 40% water, 60% protein. So that's important to know. I think in lab they actually, I don't know if you guys talked about this or not, but it may have, the numbers have been switched. Know these numbers for this course. 40% water, 60% proteins, and that goes along with the textbook that I gave you guys to read, which hopefully will be uploaded tomorrow at some point. So what are the proteins? Proteins are in alpha, beta, and gamma forms, and also GSH in high concentration. So we're going to talk about that in just a second. If you take a look down at this picture here, this is kind of a side view of the eye. Like if we took an eye, we kind of held it in our hand, and here is the cornea up here pointing upwards. We would have the anterior chamber, and then the iris, the pupil, and then here's our lens back here. So anterior cortex is here, posterior cortex. We're going to talk about all the parts, but the lens is mainly composed of a capsule, a cortex, and then a nucleus. It does have a little bit of epithelium around this section here. Okay, so what is GSH and why do we care? GSH is known as glutathione. It's a very important molecule that resides in the lens. It's basically an antioxidant, and so what happens is that UV radiation is coming into the eye, or not radiation, UV light, is coming into the eye, and that reacts with certain other molecules in our body, and then we know that that creates reactive oxygen species, or free radicals. And so glutathione because we have so much light and UV coming into the lens, the glutathione basically uses thiol groups to reduce disulfide bonds in chemical reactions, and this will essentially reduce the reactive oxygen species to a form where they can't cause damage. So this is mostly in the lens epithelium. Now if you go back to the picture from the previous slide, you can see that the epithelium is actually on that front surface, so that makes sense. Light's going to be hitting that surface first, so we've got all of this glutathione in there to basically capture any sort of free radical formation. And here I just have listed at the bottom the 3 major functions of the glutathione in the lens. So, and this is outlined in you guys' book. Maintenance of protein sulfhydryl groups in the reduced form. So here at the bottom, I've just put the glutathione molecule, and you'll see the thiol group, and so this will actually form a bond, and it won't be reactive anymore. It won't or the reactive species won't be reactive anymore. Protection of oxidation damage by detoxification of hydrogen peroxide. Removal of xenobiotics by conjugation with hydrophobic compounds having an electrophilic center. That probably sounds like a lot of garb to you guys, but it is exactly how it's written in the book. So think of maintenance, protection, removal of bad things. So just know that this is an antioxidant. I don't want you guys to get too hung up on all the specifics, but I did want to explain what it was. If you're a biochemistry buff, then maybe you'll kind of relate to this a little bit more, but I don't want you guys to worry about it as much. If you look over here where the eye is accommodating, we see that the ciliary muscle is contracting. Therefore, the suspensory ligaments are slackened. Be careful, this is kind of counterintuitive. But this allows the lens to bulge or thicken. And so it's thickening and the diameter is decreasing. So I want you guys to keep those 2 terms straight. There's a diameter and then there's a width. Okay. Also, obviously the lens, I think we talked about this in our UV chapter, it's the main UV filter. So it only allows 50% of UVR through 390 nanometers and none at 360. So if it gets 360 or lower, it's completely blocked by the lens. And so if it says it becomes opaque, that means it's completely blocked. So for, that low of wavelengths and it only allowing 50% through that 390, that's pretty good. So we're not getting a lot of that UVA and UVB into the back of our eye. Okay. So this is a quick note on embryology. I don't want you guys to get too lost in embryology. That was a topic that Alice and I actually had to throw out. We couldn't cover it. But I do want you guys to kind of be aware of how the lens forms, and there could be, a somewhat general question on this. So the lens placode, which is kind of just the development and the formation of the lens, is noted at about 22 days gestation, so in the womb. Which is crazy. That's really early. Okay, so a lens vesicle forms. Cells on the posterior wall of the lens elongate forward. Okay, so they're moving anterior. They lose nuclei, and then they become transparent lens fibers. I don't want to really get into the details other than that is how the fibers come to be. Okay. Now we're going to break it down into, there are 2 types of fibers. So we have primary lens fibers, we have secondary lens fibers. The primary lens fibers are also known as, or referred to as, the embryonic nucleus. So this is going to be the most inner portion of the adult lens. I'm going to show you guys a really good picture of this in just a second. The secondary lens fibers, also known as the fetal nucleus, are kind of the new fibers that are added and fuse around to form the Y sutures. We're going to talk about those as well. But know that think of an embryo kind of comes before a fetus, so the embryonic is more inner than the fetal. Okay. New fibers are continued to be added throughout life. They're produced by mitosis in the equatorial region. Okay, so this is kind of important. Like, where is mitosis happening? Okay, it forms the cortex. And then nuclei in this region kind of form an arc-like pattern, and they sometimes call that the lens bow. So if you see that on a diagram, that's what that means. Okay, so this is a really good slide. This is a really good picture for you guys just to kind of figure out, what's going on. So at the top of my slide is basically where the cornea would be. That's anterior. That's where the eye is pointed. Okay, so you can see where it says anterior capsule. Okay, posterior capsule, and the capsule is exactly what the capsule says. It holds and encompasses the lens. Okay. Inside the capsule, just at the anterior side, we have the lens epithelium. Okay, so this is where I said super high concentration of that GSH, glutathione. Okay, so light's going to be coming in from this direction, from up at the top. It's going to be coming into the lens, and any free radicals that are formed are going to mostly be taken care of by that glutathione. Okay, over here, so notice that I have two lines drawn. One is an equator, which is drawn basically along what we call the diameter of the lens. So equator, diameter, I don't know if you guys want to equate those two terms similarly. And then you have the axis. So anytime we talk about the axis, the pupillary axis or the visual axis, this is like along the line of sight. The axis kind of coincides with the width of the lens. So this is the part of the lens, this is in the direction, when we talk about the lens thickening, we say that this part thickens in width, and then the diameter decreases. So it gets essentially squished from here and here, from this right and left sides, or top to bottom, if this was rotated in its actual anatomical orientation. Okay, so notice that in these equatorial regions out here, we have the active mitotic region, which I just talked about before. Sometimes they call this area the lens bow, blah, blah, blah. The cortical fibers, there's going to be the cortex, which is going to be, we said capsule, cortex, nucleus. Okay, so nucleus. And then the primary fiber cells are known to be the embryonic nucleus, and the secondary fiber cells, okay, are going to be more outer. Those are known as the fetal nucleus. Here I have, we'll get to the Y sutures in a second. Here I have written, we go from the capsule to the epithelium to the cortex to the nucleus. So that is if we came in from light, or with light, to the front of the lens, and we're just going to travel through, we're going to hit capsule, epithelium, cortex, and the nucleus. Okay. Here I have a picture. This is actually not what it looks like in real life. They're much less defined, they're clear, but this is just so you guys can see what the suture looks like. So again, this picture is actually of a cataract. It's not the normal person, okay, but it does demonstrate what the sutures look like. So here, basically on the front surface of the lens, you have a suture where those fibers fused and it forms an erect Y shape. This picture is of the front of the lens because the Y is erect. If you were to change your light illumination and highlight the back of the lens, that Y actually goes upside down. It's not the same Y, it's a different Y. It's the suture that's on the back, and so it is actually inverted. So if you were to draw an upside down Y, that's what it would look like. Okay. So embryology remnants. This is basically when the lens is developing, it's going through development, all these changes are happening, the tissue's moving around. I mean, the whole eye literally is forming. And so there's some remnants that kind of may persist in some patients. Everybody has these structures, but they're supposed to essentially go away and regress. So there's something called the hyaloid artery. I don't know if you guys have heard of it yet, or Allison's talked about it in one of her classes, but the hyaloid artery is basically going to travel through the hyaloid canal. It's sometimes called Cloquet's canal, and it travels up and essentially feeds the front of the eye, the lens. It supplies nutrients to all these front structures until the eye has fully formed and it can kind of go away. So we call basically the two ends of this hyaloid artery in its canal. If it persists, you can see what's called the Mittendorf's dot, which is on the posterior surface of the lens, and that's what this picture over here on the bottom right is showing. And then if you actually have it back towards the optic disc, it's called the Bergmeister's papillae. So it actually extends from the optic nerve head. You can see on this picture, it's kind of this little fuzzy looking white kind of flap almost, just like it's kind of floating out there in the vitreous. And it's actually attached to where that hyaloid artery was coming through. So it's kind of just like a little sheath that's left over. Okay, so metabolism of the lens. So the lens is actually pretty metabolically active. There's a lot going on. So in the epithelium, it's a little bit different than where it is in the fibers. So understand that there's a difference. In the epithelium, you have, it's epithelium. Those are cells that kind of have their own little thing going on. About 50% of their energy is produced by oxidative metabolism. So if you think back to biochemistry or biology, when you guys learned the big ATP and that whole process, when you get to the end, in that channel is shooting out all of that ATP. I think it's like 32 per 1 molecule of glucose. That's going to be what the epithelium does. So very efficient, lots of energy. 50% of the epithelium's energy is produced by this oxidative metabolism process. So if we're talking about the fibers though, however, it's primarily anaerobic. What does that mean? No oxygen. So it's the same thing as glycolytic pathway. So you can think of glucose, pyruvate, lactic acid. It only makes 2 molecules of ATP versus the oxidative, so it's anaerobic. So know that those are different between the fibers and the epithelium. Know that the metabolism of the lens is pretty crazy because the lens is avascular. That makes it really hard because we don't have oxygen being carried in the blood, and so all of the oxygen and glucose and the nutrients that the lens needs is going to be coming from the aqueous. And the lens is suspended in that aqueous fluid, aqueous humor. And it's got to be able to collect. I have this picture over here just on the bottom, just kind of demonstrating how it's going to get all these nutrients in. So the lens overall, if you're on the inside of the lens, is low in sodium and high in potassium compared to the aqueous and vitreous. So this whole proportion and maintenance of how the lens kind of keeps these forces and keeps these ions in check is determined by the way that it's been set up as far as how much sodium, how much potassium. It's going to create sort of a imbalance that's on purpose, and so therefore fluid is going to move mainly from the lens to the aqueous, so from the lens out. And this is also facilitated by the gradient NACA pump. NACA is just the sodium potassium ATPase pump. It's the one that requires energy. Those are therefore found in the epithelium because they need ATP in order to work. So that's just a little bit about the metabolism. Okay, so now we're going to talk about how the lens structure is organized. We kind of already talked about it a little bit, but first we're going to start by going over the capsule. So the capsule is the outer portion, kind of holds the lens together. It's modified basement membrane. I don't know if you guys have read the collagen packet yet, or not the packet, but the text. I feel like a couple people did. You guys really need to look at that. We will have some questions on the final regarding different collagen types and so on and so forth. So because it's a modified basement membrane, it's actually going to be collagen type 4. Okay, so collagen type 4. The capsule, functions of capsule, it serves as a barrier, but it does allow diffusion of small particles because, like we said, the lens is avascular. It needs to get things in and out. It serves as a solid attachment for the zonules of Zinn. Again, that's also the same thing as the zonular fibers, the suspensory ligaments, the zonules. It's all the same thing. It creates annular thickening on the anterior and posterior surfaces. It doesn't have elastin, but the collagen is what provides flexible properties, so it can, when it accommodates, it can thicken and flatten and all this and that. The thickening rate is about 1.2% per year until the age 75. So if you guys remember back to previous lectures where I've talked about, they used to think, old ODs may still think this, that the reason people get presbyopia when they turn 40 and they've got 40 when they turn 40 and they have to use readers and so on and so forth, ODs have told patients that it's because the muscle that causes their eye to focus has become lax. So, and they're talking about the ciliary muscle, and that's actually not true. There's nothing wrong with the ciliary muscle. They've actually, Dr. Adrian Glasser has found through his research, and if you get to UHO, then you'll get to hear some lectures from him on accommodation. It's actually not that the muscle is messed up, it's actually that the lens just gets very hard. It thickens over time, it loses its flexibility properties, and so this is what essentially causes them to not be able to focus anymore. Okay, so the cortex and the capsule are actually what's thickening with the age. The nucleus is not. So keep in mind the nucleus stays the same size. Okay, so I made this note on the bottom right, where's the epithelium, just to make sure you guys understand where it is. It's inside the capsule, and it's only along the anterior side. Okay, so it's just that anterior portion of the lens, and it is held within the capsule. After you hit the epithelium, then you're going to hit cortex. Okay, so keep moving on with organization, the fibers. So that's the division between the cortex, that's the division between the capsule and the nucleus. The division between the cortex and the nucleus, however, is arbitrary. So it's not this defined like, okay, we're cortex and now we're nucleus for sure. It's kind of just meshed all together, so keep that in mind. Secondary fibers make up the majority of the adult lens. So we said that those are from the fetal nucleus. So that's the major mass of the lens. Okay, so some more numbers for you guys. Each fiber is about 2 micrometers by 10 micrometers in width, and they're about 7 millimeters or longer. So again, 2 by 10 and about 10 millimeters long. So let's think about, 7 millimeters long. So 2, 10, and 7. Okay, so 7 millimeters long, that's almost a centimeter. So when you hear these numbers, try to make sense of them. Try to think about, okay, can I relate that to something of an actual size? And that's about how big the lens is. Okay, so I don't know if you guys have talked about junctions and physiology or maybe any other classes that you guys had in your undergraduate career, but there are different types of junctions. One of the ones that is very important in the lens is called a gap junction. Its function in the lens is just to support the uniformity of the lens. Remember we said that for structures that are clear, like the cornea and the lens, we need the fibers to be very organized. We need them to be spaced correctly, we need them to be even, and therefore it'll be a transparent structure. The gap junctions also facilitate metabolic exchange between cells, so they fill the gap. Think of the gap junctions are there because if they weren't there, there would be gaps. Just think of it that way. Think of it, it's got to be there to communicate. Gap junctions are all about communication. So it's communicating the metabolic exchange between the neighboring cells. Okay, other junctions that are present that are maybe not as important as the gap junctions are the zonular adherens. Those are between the epithelium and the lens fibers, kind of keeping it together. And also interdigitating ball and socket joints, and those are mentioned in the text, and I guess they're important to some degree as far as permitting the lens to change shape. So you can think of a ball and socket kind of, it's tight, it keeps its shape, it keeps its form, but yet it's allowing this movement in a somewhat controlled environment. Okay, so lastly we have the zonules. So the zonules in, aka the sensory ligaments, I've already said all the synonymous terms. They're made of glycoprotein. Okay, so they attach the ciliary body processes to the lens. Remember that the ciliary muscle is connected to the ciliary body, and then when that pulls, when it contracts, then it's actually going to relax the fibers. So this is a great picture. You guys need to understand this picture because these are board questions. These are going to be questions on my exam. This is important to understand. So, and it's kind of counterintuitive. So, the muscle contracts, but the fibers relax. The lens thickens, the diameter decreases in the accommodated state. Okay? When you're unaccommodated, when you're looking in the distance, you're not using any of your vision to see up close, the ciliary muscle relaxes, it's not contracting, it doesn't want to accommodate. The zonular fibers are then going to be under tension. They're going to be pulled taut. Think of a trampoline. Think of those springs that pull taut and hold that trampoline in place, okay? And then therefore the diameter increases and the lens flattens. So those are the two things that you guys need to get straight. Okay, this is the graph of presbyopia. The course that presbyopia is going to eventually take. Presbyopia is mainly hit by most patients starting around 40. Age 40 is basically what we consider emerging presbyopia to kind of start. People start coming into your office and saying, I'm using readers all the time, I need help, or I want progressives. Up here at the top I have labeled the AA, which is our shorthand for amplitude of accommodation. So that means our total amplitude, our total amount that we can accommodate from birth to age 50 is a range of 16 to 2. So we start out with 16 diopters, full amplitude of accommodation. We go down to about 2 diopters, so which is essentially nothing, which is essentially why you can't read up close once you get to be 50 or older. Okay, so this again just shows the accommodation in diopters along this left-hand side. You can see the decline, and then it just goes down. Okay, so that was the lens. I highly suggest that you guys go back and read the text. I'm going to try to get that uploaded tomorrow. It's very important to kind of read these chapters because I tried not to get too much in detail during the lecture, but I think it would be very beneficial for you guys to go back and read some of this on your own. Again, it is important that you guys read the text. Okay, the vitreous. So the vitreous humor is the largest organ of the eye, and that makes sense because it's that jelly liquid that basically fills that inner portion. Okay, it makes up about 80% of the internal volume, so 80%. It's about 15 to 16 millimeters in length, 15 to 16. Its index of refraction is 1.3349. It transmits 90% of light in the 300 to 1400 nanometer waveband. Okay, so this makes sense because it's essentially mostly water, which we'll get to in a moment, but the vitreous shouldn't really have a lot of UVA and UVB. By the time light gets to the vitreous, it shouldn't have a lot of it left. So light's coming in, cornea takes a big load of that off, it absorbs, lens absorbs, and what is left, what is transmitted, which it continues to pass through the ocular media, there shouldn't be a lot of UVA and UVB. It should be mostly of the visible light spectrum, okay? So that's why that number is so high. It's because the vitreous isn't supposed to absorb a bunch of light, okay? It should have already happened by this point in the eye. There's 3 components to it. It's about 98.5% water, 98.5, okay? Collagen type 2, that's collagen type 2, and then it also has hyaluronic acid. I don't know if I say that correctly, but hyaluronic acid. And actually, I have this bracket over here. That should be extending up the collagen and hyaluronic acid. I'll get to this in a second, but I just wanted, again, the collagen and hyaluronic acid, which is abbreviated sometimes as HA. These two components interact, and the consistency of the gel, of the vitreous, is dependent upon these interactions. So it's mostly water, but yet it has this little bit amount of these, the collagen and this acid, which we'll talk about that in a second, that essentially causes it to become a gel form and has a certain consistency to it. So hyaluronic acid is a GAG. I don't know if you guys have heard of GAGs in biochemistry. That's the shorthand for it. It's essentially a glycose aminoglycan. Okay, so that is essentially sugar and protein. So it's a polysaccharide that's highly polar, thus attracts water, thus serves as a shock absorber and lubricant. Okay, so this is really important in the vitreous. So think about, you have the sclera. That's a very, durable structure. We talk about like things not penetrating, but it's like, a hollow essentially ball is only, has so much strength. And you think about a basketball, trying to pierce through that, and then it actually indenting in and then staying indented. Okay, the fact that this actually has a shock absorbing quality to it, the vitreous, which fills the inside, think of the vitreous filling inside of the basketball. When you punch that basketball, let's say if it was filled, and let's talk about a basketball that's maybe old and, not like a brand new one that you bounce it and it bounces right back up, but, one where we could press that ball in, this old basketball that usually, like let's say it's got a couple holes in it, and then you press it in, and then usually it would kind of stay indented. Well, if it has a shock absorber inside it, you're going to try to punch it, and then it's going to bounce right back. It's not going to stay indented. And so think about kind of trying to relate these functions of these different components of the eye. I mean, the vitreous and the lens aren't the most exciting parts of the eye, but they are very important. Okay, keep going. The three origins of the vitreous. So this is something that kind of talks about in the book. It's kind of abstract sounding, but it is on boards. It is important. They talk about the primary, secondary, and tertiary. Primary is essentially an extension of the hyaloid artery adventitia. So I talked to you guys about the hyaloid artery during development. It kind of supplied the anterior structures of the eye. Well, in arteries, there's a layer called the adventitia, and from that is where the primary vitreous essentially came from. And the primary vitreous is vascular, okay? The secondary origin of the vitreous is basically extracellular matrix consisting of type 2 collagen. It essentially pushes the primary forward, and it's avascular, right? Because we already talked about the lens and the vitreous are avascular structures. And then the tertiary origin is going to be the zonular fibers suspending the crystalline lens. So if I ask you guys, where do the zonular fibers come from? They don't come from the lens, they don't come from the retina, they actually are an origin of the vitreous. Okay, so here's actually a really neat picture. I think I have it blown up a bit, so we'll go over that in a second. Okay, perfect. So, if we take a look at this picture, I've kind of outlined in pink what I feel like is important for you guys to know. So here we have that when the eye is developing, we have that kind of pathway called the cloaca's canal or hyaloid canal. This is where the hyaloid artery comes forward and feeds the anterior structures. It forms a space when it gets to the back of the lens. Remember we had that remnant, which was Mittendorf's dot on the posterior surface of the lens, but this space right here is actually called Berger space, so the retrolental space. So retro meaning behind, lental meaning lens. Okay. Secondary vitreous, that's what we said was the portion that kind of pushed forward. So we had the primary vitreous was a remnant of the hyaloid artery adventitia. We have the secondary vitreous, the tertiary vitreous, which is our zonular fibers, and then I don't know if I want to talk about that yet or not. I guess I want to kind of just make a point here about the vitreous space. We're going to talk about where the vitreous is tightly adhered to, or how is it attached. It is a gel, but it does have attachment, so it stays. It's not constantly shaking around. It's somewhat stable. Also, I have kind of circled right here is Weger's ligament. So this is just a remnant on the back of the lens. This is another, I'm sorry, it's not a remnant, it's an attachment, and we'll get to that in just a second. I also want to talk about, down here I've just kind of made a point that the vitreous has an outer layer referred to as the vitreous cortex. The density of collagen here is thicker than the inner vitreous, and the hyocytes are more prevalent. The thickness, we say it's about 200 microns. There is none over the optic disc. So know that there's kind of an outer layer, and this isn't demarcated on pictures and things. Just know that in this jelly-like fluid, the outer portion is known as the cortex, and it's a little bit thicker because it has more hyocytes, and then the inner is a little bit less thick or less viscous. Okay, here are the vitreous attachments. So these are very important to understand. Why? Because patients can have detachment. Now, what you guys have probably heard about for the most part is retinal detachment. Vitreal detachments can cause retinal detachments. Why? Because they're closely adhered. The vitreous is right up against the retina. And so sometimes, I don't know if you guys have heard of vitreoretinal traction, vitreomacular traction. If you're looking on OCT and things, you can see where essentially these bubbles or these masses are kind of, it's like pulling up a sheet of, it's like pulling up a sheet on a bedspread and then it not laying flat, or it's like, kind of carpet that kind of starts to get wrinkled in some spots, it starts coming up off of, the ground underneath. It can pull on the retina and essentially can cause a retinal detachment. It definitely can cause some vision changes. So it is important to know where the vitreous is closely attached or adhered. So first we'll talk about the vitreous base. That's what they call it. This is the strongest attachment. It's annular, so it's around the entirety of the eye. We just talked about how the ciliary body goes all the way around in an annular ring and an annular structure. It's about 1.5 millimeters at the ora serrata. Okay, so it's the transition from the ciliary body to the retina. Right at that region, we're going to have what's called the vitreous base, and that's where it's going to tightly attach. Okay, the second one we're going to talk about is the ligament of Weger. That's the second strongest. That was back on that picture that I just showed you. It's on the posterior lens. Again, it's annular. Okay, and that's just another attachment. We also have the optic nerve head, and then also the fovea that serve as attachments. Cells that are found in the vitreous, most prevalent, we say the vitreal cells, so to speak, are basically hyocytes, and these are kind of star-shaped little cells that have kind of an oval nucleus. You can see in this picture here, it kind of shows that. And these are essentially macrophages in the cortex of the vitreous. Remember we said the cortex is kind of the outer, thicker part of the vitreous. And then they also have the highest density at the vitreous base, which is what we just talked about. What is their job? Okay, so these guys synthesize HA, which is that hyaluronic acid, glycoprotein, and collagen. So these are essentially the cells that are secreting what interacts with water and then kind of makes that gel consistency. So these cells are super important to the vitreous. Down here I just have a little biochemistry of hyaluronic acid. And then we also have fibrocytes. Those are at the vitreous base as well, and those help synthesize collagen. Glial cells, this is kind of debated. They said that they're in the perimeter, they're there. I think it's debated on what the function is as well. They're said to be ectopic. If you guys don't know that word, that means it's essentially somewhere where it's not supposed to be. So if you talk about an ectopic pregnancy, that's a pregnancy not happening correctly in the uterus. It's not in that location correctly. It could be somewhere else. An ectopic tumor, you could have all sort of ectopic things. It means it didn't really originate there. It's not supposed to be there, is what ectopic means. Okay, vitreous metabolism. So even though it's avascular, it's very metabolically active, okay? There's a lot going on in the vitreous. It's very important for the eye. I thought this picture was cool off to the right with just showing an eye sitting there, of course in black and white, and I don't know if the replica, I'm not sure exactly. I just liked the picture. So in the vitreous, oxygen tension is lowest centrally. That's just a little factoid that you gotta know. 50% of the water is actually turned over in 10 to 15 minutes. That's important to know. It has to maintain a barrier to cell invasion to maintain transparency. So again, light is still traveling to the back of the eye. We haven't reached the retina yet, so it's important that everything's transparent. Vision isn't being inhibited by anything that's floating around. Mechanical support to surrounding tissues, okay? And then also the viscoelastic properties, being a shock absorber, which we already talked about with the basketball analogy, which I don't know if that was the best analogy. I'm sorry if it wasn't, but I'm sure you guys can kind of talk about it amongst yourselves and come up with something to kind of understand what I'm talking about. Okay, this is an important chart to know. So basically when we are born, the vitreous is only a gel, and then as we age, it becomes liquefied due to the rearrangement of collagen fibers. So you can see these numbers on here, and this is totally fair game for me to ask you guys a question. I would just memorize writing this chart. We had to know it for our class. You'll probably have to know it for board. You can see down here at age 65, 60% liquid and only 40% gel. So again, as we age, things just kind of start going down. This is a picture of a PVD, a posterior vitreal detachment. I thought that this was really relevant because people come in with these all the time, especially older people. So if you guys have ever heard of that before, just a little bit of clinical something just to make it relevant to you guys. Basically what happens is, like I was saying, kind of we call it a vitreal retinal traction. Basically, the vitreous cortex separates from that the inner layer of the retina, which is the inner limiting lamina. The retina has many layers, and we're going to talk about that when we get to it. And I know Allison's going to talk about the retina next week with you guys. But what happens is you can see down here in this picture of an OCT, you can see here we have the fovea. We have all of our retinal layers, and this is the vitreous, and this is that kind of inner limiting lamina of the retina, and the vitreous has pulled it, and this has formed basically a pocket. Okay, it's weird because there's not really a decrease in vision, but because there's been this pulling sensation, things are kind of shaking around. Patients are going to report seeing floaters, which are like little cells kind of floating around in there. It could be a little bit of pigment. It affects about 65% of the population over the age 65. So, again, very relevant. Makes sense to know about this for your patient. This is just a BIO view over here on the right-hand side. The background looks blurry, so the back of the eye with the optic disc and all of that looks blurry because we're actually in focus on this film right here, which is where the vitreous has essentially come unattached from the retina.